All right. Condensation of a dipeptide means you take two amino acids and you put them together.
C. There's C, and then there's the O.
Hey, Kyle, where the bio teacher?
Cool. That's one amino acid.
Oh, wait,
Here's the other. For like annotation purposes, if you had to write like an essay about this instead of drawing it, you could refer to some specific parts. This is called the carboxyl group. This is the amine group. And for the record, just in case you're wondering, an OH is called a hydroxide. And just for kicks, as long as we're going over them, a C with 3 H's on it, but it is connected to something else, is called a methyl group. But that doesn't exist in an amino acid, so I wouldn't worry about it too much. But C with 3 H's, that's always a methyl group. So, how do we put one of these puppies together? Well, you take the hydroxide group from a carboxyl group and a hydrogen from an amine group, and both of those will become an H2O water molecule, which is why this is called condensation. A water molecule is formed. In that sense, you join the carboxyl and the amine groups in a beautiful union. And that is a dipeptide with a peptide bond.
Isn't a dipeptide bond theoretically 2?
It's not called a dipeptide bond. It's called a dipeptide because there are 2 amino acids. A similar example would be if you were to take 2 glucose molecules and put them together into a disaccharide. That would just be 2 sugar molecules linked with a glycosidic bond. So I'll just review those real quick. Glycosidic is for saccharides. I think there's another C in there. Does anybody remember what it's called when you put a triglyceride together, what those bonds are?
No.
It's an ester bond. Which is, we have a student in the junior class named Esteban, and everybody just calls him Esteban. And that would be in a triglyceride. And just so you guys kind of see what the triglyceride is all about, this one does pop up every so often. So, that's pretty good. I want to show it. Here we go. So, chill out. This is a fatty acid, long chain of carbons. This is a glycerol, 3 carbons with OHs. Again, you take out a water molecule and you just join them into a triglyceride. So sometimes the question's like, how many fatty acids can you put onto a glycerol? 1, 2, 3. But I don't think you'd ever need to draw that one. You might just need to identify a glycerol or maybe a fatty acid with a methyl group on the end. Luca, you had your hand up? Or was that
me?
I just saw a hand, sorry. Yeah.
So on the peptide bond, the oxygen on the left side, does it matter if it's above the C or below the C?
I think theoretically that doesn't change the molecular structure to flip them, but I would stick with this.
Okay.
I would stick with this convention. And then I would keep these H's and O's upright.
Okay.
Cool. So much we go over in this class. So many things we learn.
That's actually like what I'm saying about it. Like we learned like a couple of stuff in our teeth.
You wouldn't get it.
Like
Joke.
for
Don't
shared
get the
content. Here it is.
Yeah. There it is.
I know that's so many.
Okay. Enzymes, DNA. DNA it is.
Yeah, it is.
Man, I hate
Gonna need to draw the whole like.
No. That was a learning diagram. All right.
It was awesome.
We're gonna do a teeny tiny DNA molecule.
Wait, I remember this.
What we have is a nucleotide. The nucleotide consists of a nitrogenous base. This would be either A, T, G, or C. This is a ribose sugar. Anybody remember what this one is?
It's a phosphate.
It's a phosphate. And if you put 2 more phosphates on it, that would be very close to an ATP molecule. Now, when you draw these, it is important the way you bond them. So, if we're numbering our carbons, this is an oxygen up here. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 is up and off, connected to the phosphate. You need to connect carbon 5 and carbon 3. The nitrogenous base is always connected to carbon 1. And I'm not going to go crazy here. I'm just going to put in one more row of nucleotides. And I'm going to connect them.
And what is the actual ATGC made of?
The nitrogenous bases? Yeah, a nitrogenous base. On the other side, your nitrogenous bases are flipped 180 degrees. So, make sure that the bases are still touching each other, but now your pentagon should point down. And the phosphate goes down and to the right, whereas this one went up and to the left. Add a couple more in. And I have done prettier diagrams in my life. Okay.
This is
Part of being a teacher is accepting that perfectionism is no longer an option. I got over that one pretty quick. Connect them. Here we go. Let's just say we had an A. What would that bond to?
T.
AT. How many hydrogen bonds does AT have? It's either 2 or 3. 2. 2 dotted lines. G and C will have 3. And from there are a whole bunch of things that you can label. I'm going to just put up the list so that you can kind of cross-reference that list against what you've got drawn. And more than knowing, more than labeling all of these things, it's more important that you just know what they are and why they're there. So, the double helix. When we drew this one in class, we did a little one of these, and we just connected it across.
Well, neato.
And just know that the DNA is called a double helix when there's 2 strands. The major and the minor groove, not super crucial, especially not in the diagram, but there's one side of the helix where the groove is a little smaller than the other side. But here's where the real meat starts. A nucleotide is right here. This, for the record, I'm going to do one that is not a nucleotide. This is not a nucleotide. That is part of one nucleotide and then some of the other. Base pairs like G and C. Hydrogen bonds with your dotted lines. 5 and 3 ends. Where is the 5' end up here?
The top on the
With
left,
the
top
5
left.
carbon.
By the 5th carbon. 5', and then the 3rd carbon right here gives this the 3'. notice that you flip them on the other side. I took it off the page, but this would be the 5' and this would be the 3' end. DNA is always read and constructed from 5 to 3. So this strand would be read A, G, whatever. This strand is read the opposite direction, something CT. Bases, hydrogen bonds, 5' ends. Anti-parallel. They're called anti-parallel because when you're reading or making them, you have to make one this way, and the other one this way. Sugar phosphate backbone, that is the sugar and phosphate backbone over here. It has 2 of them. And it's also worth remembering that one of the strands is the coding strand, and the other strand does nothing. It stabilizes the molecule. It provides a basis for replication, because if I gave you the non-coding strand, you could give me the coding one, right? Base pairing dictates that. Cool.
How would you spell the sugar phosphate backbone again?
What I did is, in class, I just did one of these, and then labeled it the sugar phosphate backbone.
And how would you label the coding and non-coding?
Oh, and I what?
Label the coding and non-coding strands? Would you just draw lines and write that?
I don't think they would ask you to label anything like that. I put that on this diagram because it's a great summary of all things DNA. I don't think you'd label a coding or a non-coding strand. But you'd need to know that one of them is coding and non-coding.
Can you, sorry, can you go over coding and non-coding?
Sure. The most important way to go over coding and non-coding. Here.
Is it just like one of them codes or one of them doesn't?
So here's the important thing. If you're heading out, happy studying. Thanks for coming by. So, very briefly, in transcription, when you make the mRNA, you build it complementary to the non-coding, or what's called the template strand, so that it is the same as the coding strand. That's what's up with coding and non-coding strands. This, if you read it, if you turned all of these into codons, and you used that little codon chart to put an amino acid together, these are the instructions for a protein. These are BS. These are not going to get you anything.
Okay. And is it, is the the one that is easier to read so you don't have to use the Okazaki fragments, is that always the non-template strand? Or
Nope, it just depends on which direction replication is proceeding. Because replication starts in an origin of replication and moves both ways apart, and it forms this replication bubble. If you remember.
Oh yeah.
And on that bubble, one is a lagging strand, but later on it's a leading strand, and one is a leading, and later it's a lagging. So the same strand could be a leading and a lagging strand depending upon where the helicase is. Yeah?
So, in terms of the strand that isn't doing anything, you build the other DNA against that, and that's how you get it?
You build mRNA complementary to the non-coding strand. But what that does is complementary to the non-coding strand is the coding strand. So what you want is mRNA that has the same code as the coding strand.
Oh, okay, I get it.
The only difference is that T's are now used. Because DNA does not have a U or a uracil. Alright. Alright. You ready for a 5-minute break?
Yeah.
Alright. I could go all night.